Demographic and evolutionary responses to stochastic environments.
All right, so can you see my share?
Yes, we can.
Yes, okay, so hello everyone, so thanks for having me. A bit like Claudia's talk yesterday, I tried to pack different projects in the same talk to make a more coherent story, so I hope I can stick to the time, and otherwise I just have to, I guess, pardon myself in advance, since this is the new fashion, it seems. So this is about demographic and evolutionary responses to stochastic environments. And the type of observation that stimulates this work is that when you look at about any time series of natural environments, you can see some tendencies, such as this global warming on this global temperature data sets, but you also see lots of noise around these tendencies. And this noise, this random noise, is usually faster than the trend. So it's the major stress that organisms are facing in the wild. And we know that global change is also altering these patterns and the predictability and the magnitude of these fluctuations. And this type of environmental stochasticity has demographic consequences, because obviously the environment affects demography, the survival and fecundity of individuals, which translates to fluctuation in the population size or density. And when you look at time series of abundance, you almost always see these types of sometimes large fluctuations that are not explained only by internal feedbacks. So it's a strong source of stochasticity, because this environmental stochasticity, unlike demographic stochasticity, it may place an initially large population at risk of extinction. A bad year is a bad year for everyone, so it can cause large declines. For evolution, of course, this is a source of fluctuating selection, whereby different genotypes or phenotypes are favored by natural selection in different times. So if you think of an emblematic example, such as this phenological trait like breeding time in these blue teats, if you track them across here, selection on them changes in magnitude and sometimes in sign too. And since this is stochastic variation in selection, this is a source of chance in evolution, just like drift. This can increase the variance among replicates. If you're trying to project what's going on, you need to account for this envelope of uncertainty. And then the questions become, to what extent can we predict and understand these ecological responses and evolutionary responses to stochastic environments? And also, can we rely on simple and general frameworks for doing so, or do we always need the more type of specific mechanistic models? So I would try to argue for the use of simple models and see how far we can take them. So one basic principle that we can rely on is that evolution and demography are connected through the fitness landscape. By this, I mean the relationship between fitness or mean fitness and genotypes or phenotypes. So if you take the simplest discrete type model, where you have, for instance, the multiplicative fitness is W for an individual, and the mean fitness is W bar, then demography, the population size at time t+1 is equal n times W bar, right? So you can write this on the log scale, and then it becomes additive. So you encounter this log W bar. And when you're interested in evolution, the change in allelic frequency caused by selection depends on this term, which has to do with heterozygosity or genetic variance at that locus, and this derivative of log fitness. And actually, the same can be done for quantitative trait, where you have this derivative, but with respect to the mean phenotype. So you encounter this log mean fitness in both, in all this, in ecology and evolution. And this here is called the selection gradient. Another kind of basic principle is that adaptation is thought to be a match between an organism's phenotype and the environment. And this is often conceived in verbal model and model explicitly as an optimum phenotype that moves with the environment. So you can think of this relationship between the phenotype and fitness that has an optimum in each environment, and then this optimum is changing with the environment. So this is in many, many models of adaptation to changing environment reviewed in here, for instance. But you can also sort of demonstrate this in natural population. This is what we have done a couple of years back in this example of a breeding time in great teats, and we were able to estimate the location of the optimum and these patterns of fluctuations over time. So this is the black line here, and the mean phenotype is are the dots. There are also lines of evidence coming from distribution of fitness effects of mutations, but I won't go into them here. It has to do with Fisher's geometrical model. Perhaps you'll hear about that from Guillaume Martin tomorrow. One last point that's important to consider biologically is that for many organisms and many traits, the phenotype of a given genotype may change with the environment as a result of phenotypic plasticity. And this means that so for instance, if you stick to this example of phenology, so these are here different populations, but this is also found at the individual level, that individuals breed earlier in warmer years. So there's a reaction norm of how the trait changes with the environment. And when you combine these reaction norms, this plasticity of different genotypes in response to the environment, with the fitness landscape with a moving optimum, you end up recovering this type of tolerance curve with fitness against the environment, which are observed especially for many microbes where you don't necessarily have the underlying traits, you just measure fitness component. Then you end up with this tolerance curve. For instance, this more plastic red genotype tracks the optimum more closely, and then it has a broader tolerance curve. Okay, so these are just the kind of basic concepts that you can try to use to project and try to understand what's going on in some kind of general way. And now the question is, how far can we take this type of moving optimum theory to try to account for population and dynamics and population genetics in randomly changing environments? And especially, what can we learn from trying to contrast such simple models with laboratory experiments where you control this pattern of fluctuations experimentally? And so the common assumption to this theory and the experiments would be to assume a Gaussian process in the environment. So there's a Gaussian distribution of the environment in the long run, but then the environment might be autocorrelated. So if there's weak autocorrelation, then the fluctuations are very noisy and erratic. And if there's a large autocorrelation on one time step, then the time scale of fluctuations is larger. All right, so in terms of experiments, what we have used is this halotolerance microalga, Dunaliella salina, which can thrive from almost fresh water to saturated brine, where it becomes orange because of producing carotene and contributes to this spectacular pictures that you may have seen. But that's not what we'll talk about today. The idea is that we use this organism because it has this broad, this mechanism for tolerate a large range of salinity. And so we change salinity twice a week for several months. So this means hundreds of generations for this organism. And we used this type of liquid handling robot, pipetting robot, which allowed us to do an ample replication and to have complex patterns of fluctuations with different time series for each line within a treatment. And by complex, I mean that we were able to simulate this, this type of Gaussian processes. So these are the actual time series that we use in the experiment for one line for each example. So for instance, so we went from zero autocorrelation, so no correlation between subsequent time steps, to a negative autocorrelation or positive but of the same degree, and then high positive autocorrelation. So where you see that one time step is very strongly correlated to the previous one. All right, so let's start with the population dynamics. What can you predict using these simple models? When you have a Gaussian fitness peak, then population growth rate, which depends on the log mean fitness, so it depends on the squared mean mismatch with the optimum. And you can include a density dependent term also in the model to include density regulation. And so what's important is that, so we assume that in a randomly changing environment, this maladaptation term here, this phenotypic mismatch depends on the environmental changes in theta and also in evolution and plasticity that will change this mean phenotype Z bar. So if we first get rid of this density dependent term and we think of population starting at low density, then there's a transient phase of exponential growth where the effects of past maladaptation just sum up. So you end up summing these square terms here. And when theta is a Gaussian process, the log population size small n then becomes some kind of chi-square. In fact, it's a non-central reverse displaced times chi-square with a degree of freedom that increases over time because you sum more terms. So this, from this, you have a distribution that's initially very skewed like that. It's bounded to the right because it cannot grow better than the phenotype or that would always be at the optimum. So it's skewed negative to the left. And then it's less and less skewed over time, but it can keep an excess of this excess of low population size for some time, for instance. This is one of the predictions of the models. Then beyond this transient phase, you can think of what happens in the long run. The population eventually will fluctuate around the mean carrying capacity, and you can get some analytical results for that if you focus on the type of density regulation that's convenient. For instance, this Gompertz type, which depends on log population size. If we parameterize the strength of density dependence with this parameter phi, then you end up finding that the asymptotic population size n depends on this sum here. Which what you need to notice from that is simple equation, but it's instead of just summing the past maladaptation, now it's a weighted average with more weight on more recent events. In the sense, density dependent erases the influence of past maladaptation, and as a result, the stationary distribution at stationarity it is skewed. It doesn't tend toward the normal as in the previous case. And it's more skewed under higher autocorrelation or stronger density dependence. For instance, here, this higher autocorrelation and strong density dependence, you have this skewed distribution because you end up summing effectively fewer mismatches with the optimum. So these are, and there are other predictions that I can go into here, but these are things that are quite generic in a way. And we try to look at, can we look at some of this at least? So in our experiments that I described before, we had 40 lines, about 40 lines for each autocorrelation treatment. We actually did this for several genotypes. And we also had 3 constant salinities. And what we did in these experiments is that we tracked population size at each transfer using flow cytometry and spectrometry. And so we could have this distribution of population size across all replicates for different treatments, etc. And what we immediately see is that after a few transfers, the population is no longer in exponential growth and you reach the stationary distribution. And so when we focus on this phase where it's stationary, we reach this distribution that at least graphically looks quite a lot like what we found in theory with this negative skew, etc. So, but the question is it caused by the same process that we had in mind? So to do this, we need to go to look at the growth rate, the intrinsic growth rate, and this we could get by looking at the actual time series of salinities, because we know which time series of salinity we applied, and we also have the growth rate, and we can just graphically, we can already see a few things like here the population size, the salinity is declining and then increasing, and this is causing a large negative growth rate, for instance. So when we combine all this, we can estimate actually a tolerance curve which has the growth rate against the environment. And in fact, this shows that the variation in population growth rate in our experiment is quite well predicted by this kind of tolerance curve with an optimum that I described before. Here it's quadratic and not Gaussian because this is Malthusian fitness. But however, to correctly describe what's going on, this tolerance curve has to include not only the effect of the current salinity, but also of the previous salinity just before the previous transfer. And for instance, if you look at the effect of environmental autocorrelation on the mean growth rate, the variance of growth rate, and we also look at higher moments, if you don't include this, this memory effect, you wouldn't predict any effect. You wouldn't find an effect of environmental autocorrelation, whereas you do find it correctly if you include this memory. So we interpret this as an effect of phenotypic memory caused by lagged phenotypic plasticity. The state of the cell is still responding to the environment it was in a few days ago, which is a few generations back. So I want to now move to the population genetics. Here, using this same kind of model, if you think of the simplest model where you have haploid selection on a mutation with a phenotypic effect alpha on the trait, and the background phenotype of the trait is M, and then there's an optimum theta, then it's convenient to focus on the logit of allelic frequency, and then we can show that it's depending quadratically on the effect of the mutation, but linearly on the deviation from the optimum. So mutations that compensate the mean mismatch are favored. As I said, there's a linear influence of the maladaptation, which means that with the Gaussian process of the environment, you get a logit, a Gaussian distribution of this logit allelic frequency over time. And this was already present in models by Kimura and Gillespie a long time ago, but they didn't have an explicit phenotype. They just had distribution of selection coefficients. And so the model predicts that the expected trajectory of this on the logit scale does not depend on the pattern of fluctuation. It just depends on the initial mismatch with the optimum. So when you change the pattern of fluctuations, you get the same mean in blue here. You just increase the variance among replicates. The variance among replicates, the stochastic variance of the process, increases linearly over time and faster under higher autocorrelation of the optimum. And this is on the logit scale, and when you move this to the actual frequency scale, it will transform back. This variance on the logit scale can translate into change in the timing of these selective sweeps. So when there's more variance, you go from very low to very high frequency at different times. All right, so one last bit of theory. I won't even show a graph on this, but just that mutation may also have a phenotypic effect that changes with the environment. In this case, you can think of a mutation for plasticity, which changes the slope of plasticity by a certain amount. Then different genotypes would have different environmental tolerance breadth, as I showed before. And what happens in this case is that the variance of environmental fluctuations then influences the expected trajectory, unlike what I just showed. All right, I just skipped this last line. So here again, we try to look at what we can see in our experiments. In the same experiment I showed before, but we have mixtures of two strains, and we were able to track the frequency of one of them by amplicon sequencing of a barcode sequence. And so, for instance, this is what we got for this constant salinity, so low, medium, high salinity, and then these are the fluctuating treatments, and these are, so the inboxes are the salinities time series. So if we start by looking at the mean, the expected trajectories, first thing we saw is that the expected trajectory in a fluctuating environment here in black is below that in a constant environment that has the same mean here in blue. So this is consistent with what we found if we look at the selection coefficient in constant environment. We find a concave relationship. So from Jensen's inequality, we expect that if we increase the variance of salinity, then we should reduce the expected selection coefficient. This also suggests that there is a strain difference in plasticity or tolerance breadth, because otherwise you wouldn't find this, which we found out in our model. We found that the autocorrelation treatment influences the expected trajectory in our experiment. There's a significantly higher mean selection coefficient in highly autocorrelated environment here in red, and they look a lot like the constant environment, in fact. And one last result is that the variance, the stochastic variance of this population genetics increases faster in time in a stochastic environment, which is reassuring, as you would predict, but we didn't find a detectable influence of the autocorrelation treatment on this variance. So we could only find that the treatments with variance had more their variance among replicates increases faster. So just to finish on this, the argument is that this kind of simple, plausible models with moving optimum, they capture at least some key qualitative and quantitative aspects of these experiments in stochastic environment. There's a lack of precision and replication, of course, which limits the exploration of some aspects, such as these things about stochastic variance that I talked about in the end. And what's interesting I found is that deviation from the simple models sometimes can highlight some important biology, like these memory effects for population dynamics, this difference in tolerance breadth in the population genetic example. And of course, more precise mechanistic models are really valuable, and they would be more precise to predict what's going on in the lab, to understand. But you can argue that they would be less useful for extrapolating to natural populations. So it's good to try to validate these simpler models in the lab also. To me, one of the challenges ahead is try to combine this type of external forcing, which may be stochastic as here, with internal feedbacks of the kind that we described before, like cross-feeding, etc. So with this, I'd like to thank the people who worked on this. So mostly Olivier helped with the theory, and Marie, who did most of the experimental work you've seen here, and can take questions.
Right, thank you. We do have a